Carbapenem-resistant Enterobacteriaceae (CRE) is a significant public health concern worldwide. CRE is a collective name of massively diverse group of organisms that are primarily colonizers of the lower gastrointestinal tract of humans and animals as part of their normal flora. Enterobacteriaceae family species have high level of resistance to a class of beta-lactam antibiotics, carbapenems. Food handlers, who are considered as an important link in the chain from farm to fork, as well as they are at high risk of being CRE colonized or infected. Also, food handlers play an essential role in spreading these organisms in community as well as healthcare settings. At the same time, food handlers have an important integral part in the community in preventing food contamination. This is largely depend on their health status and hygiene practices that may occur at any point in its journey from the producer to the consumer. The emergence of antimicrobial resistance as a global problem has been extensively studied in the healthcare settings. However, little is known about the epidemiology of CRE in the community, and the information is strongly needed. The overall goal of this work was to determine the prevalence of carbapenem-resistant Enterobacteriaceae rectal colonization rate among healthy food handlers working in both community and healthcare settings in the state of Kuwait, as well as the rectal carriage rate in patients with proven CRE infections as infected control patients. To highlight the molecular epidemiology of rectal isolates of CRE and identify carbapenem-resistant genes harbored by food handlers and infected control patients. To demonstrate the genetic relatedness of rectal isolates of CRE from food handlers and infected control patients. Fecal samples were prospectively self-collected by 405 food handlers, and rectal swabs were collected from 92 infected control patients. The samples were immediately cultured on appropriate agar plates. Enterobacteriaceae isolates were identified using Vitic2 ID system. Antimicrobial susceptibility testing were performed using e-test and agar dilution methods, according to interpretive criteria by CLSI. Carbapenemase encoding genes were detected by PCR and sequencing analysis. Clonal relatedness of CRE E. coli and Klebsiella pneumoniae isolates presenting for carbapenemase genes were determined by multi-locus sequence typing (MLST) and diversity lab typing using procedures were previously described. The allelic profiles, or STs, were assembled for each strain and cross-referenced with the GenBank database, then clustering STs into clonal complexes using e-burst analysis. Microbiological cultures of 405 samples from food handlers yielded 681 Enterobacteriaceae species. Of these, 62.4% were E. coli and 18.5% were Klebsiella pneumoniae isolates. However, 92 samples from infected control patients yielded 95 Enterobacteriaceae species. Of these, 72.6% were Klebsiella pneumoniae and 16.8% were E. coli isolates. The prevalences of CRE were 5.4% and 91.3% among food handlers and infected control patients, respectively. More details are shown in Table 1. The predominant CRE genes harbored by food handlers isolates were 86.4% KPC, and the detected variants were KPC 18, 2, and 29, followed by 45.5% OXA 48 and 9% NDM with detected variants NDM1 and NDM7, while in infected control patients isolates 67.7% OXA, including variants OXA 48, 181, and 232, followed by 40% of KPC, including variants as KPC 2, 18, 20, and 29, and 36.9% NDM, including variants as NDM1, 5, 6, and 7. Figure 1 shows the dendrogram of clonal relatedness of carbapenem-resistant E. coli isolates from food handlers and infected control patients using MLST. 7 isolates from food handlers were genotypically distinct, with 7 different STs. Of these, 5 were previously known: ST1876, ST295, ST10, ST1415, and ST38. Additionally, 2 novel STs were identified for the first time in this study: STN1 and STN2. STs were assigned into 5 clonal complexes. However, 6 isolates from the infected control patients were analyzed with 6 different STs. Of these, 4 STs were previously known: ST410, ST1276, ST405, and ST69, and 2 closely related isolates from infected control patients belonging to ST1276 and ST405 were obtained from a Pakistani patient admitted to Mubarak Al-Kabir Hospital in Hawali Governorate and a Kuwaiti patient admitted to Ibn Sina Hospital in Al-Asima Governorate, respectively, with the coexistence of OXA and KPC carbapenem-resistant. Another 2 closely related isolates belonging to STN3 and ST410 were obtained from a Filipino and a Somalian patients admitted to Mubarak Al-Kabir Hospital in Hawali Governorate. The other strains showed considerable genetic diversity. Figure 2 and Figure 3 show fingerprinting analysis of carbapenem-resistant E. coli using diversity lab typing. Figure 2 shows the genetic relatedness of 7 carbapenem-resistant E. coli isolates from food handlers. The dendrogram showed a clonal proximity of 95 till 97% for 2 isolates, 4E and 5E, recovered from Indian healthcare food handler and Filipino community food handler, respectively, were considered as similar. Figure 3 shows the genetic relatedness of 12 carbapenem-resistant E. coli isolates from infected control patients. The dendrogram showed a similarity less than 95%, and isolates had no clonal proximity with each other. Therefore, isolates were considered as different. Figure 4 shows the dendrogram of clonal relatedness of carbapenem-resistant Klebsiella pneumoniae from food handlers and infected control patients using MLST. 7 isolates were detected from food handlers with genotypically distinct with 7 different STs. Of these, 4 were previously known: ST461, ST268, ST25, and ST2389. 3 novel STs were assigned by Pasteur Institute MLST database with a new designation ST3495, ST3496, and ST3497. STs were assigned into 5 clonal complexes. 6 isolates from the infected control patients revealed 6 different STs. 5 were previously known: ST37, ST147, ST1880, STs 2059, and 2 identical ST231. A novel allele with a new designation, ST4743, was detected. STs were assigned into 5 clonal complexes. 2 identical isolates, K429 and K430, with ST231 from Kuwaiti patients admitted to Mubarak Al-Kabir Hospital in Hawali Governorate, harboring OXA and KPC genes. 2 closely related isolates, K467 with ST147 and K470 with ST1880, were isolated from Egyptian and Kuwaiti patients, respectively, admitted to Mubarak Al-Kabir Hospital in Hawali Governorate, harboring NDM and KPC genes, indicating inter-hospital spread of CRE isolates may have occurred at some point in Mubarak Al-Kabir Hospital. Figure 5 shows fingerprinting analysis of carbapenem-resistant Klebsiella pneumoniae isolates from infected control patients using Diversity lab typing. Dendrogram showing genetic relatedness of 19 carbapenem-resistant Klebsiella pneumoniae isolates. Clonal proximity of more than 97% of 2 isolates, 6K and 7K, were detected from patients admitted to different hospitals, Mubarak Al-Kabir Hospital and Al-Farwaniya Hospital, which were considered as parent clones. Clonal proximity of more than 90% of 2 isolates, 4K and 5K, were detected from patients admitted to the same hospital, Mubarak Al-Kabir Hospital, which were considered as related clones. Our study revealed alarming and high prevalence rates of CRE rectal colonization among food handlers and infected control patients, suggesting that the dissemination of resistant genes among individuals may occur beyond the healthcare settings and probably responsible for this dynamic transmission. These isolates belong to heterogenic genetic backgrounds, suggesting that the isolates might have been imported from diverse geographical regions. The commonest mediating gene was black KPC among food handlers isolates and black OXA types among infected control patients isolates. Close monitoring of fecal coverage of CRE by food handlers and strengthening of hygiene measures are imperative to limit their spread in our community.